from our technical conferences so that we can give everyone a chance to sign up for a slot for this round. And today we're scheduled to start at 8 and end at 9 Pacific time. These conferences are recorded, so you can access them online. If you have any questions during the presentation, please type it in the message box provided in the application. And also, please mute your mic during the presentation to prevent any unnecessary background noises. Are there any questions? Okay, so we'll start with Brian Bleakley, and he'll be continuing his discussions of ProTurn collaborations from last week.
Hi. Yeah. So last week last week we talked about ProTurn collaborations. So just a basic review. So ProTurn, like we mentioned, it's for calculating protein turnover rates using heavy water labeling. So as organisms enrich with heavy water over a period of days, it's going to be incorporated into their proteins, and that will show up in the mass spec. And using ProTurn, we can calculate each protein's turnover rate. So basically, the collaborations we talked about last week were with COPaKB. So currently, ProTurn can download and analyze COPaKB data, and we're overhauling the COPaKB API, and we'll update ProTurn as we go along. Also, the integrated proteomics pipeline, IP2, it's a commercial enterprise, but they offer a development kit which should allow us to make a plugin for ProTurn. Let's see. Matthew, are you online?
I am, yes.
Oh. Hey, Matthew.
Hey.
Hi. So you work with at the Scripps Lab. Do you work with IP2?
I do not work a lot with IP2. I mean, I mostly the people that use IP2 are the data generators, I guess, if I could say it like that. Robin is really the person that does work the most with IP2. My work focuses on developing tools around IP2, if I could say it like that.
Oh, okay. What's the plugin process like? So if we make a plugin for IP2, is that available then to all the other users?
I think it has to be approved by the IP2 people before it makes it available. You can obviously use it for yourself, but before it makes it available to all users, it has to be approved by the people there. So they review the plugins in order to not distribute something that they believe is not working.
Okay, so then we basically make our own. We clone our own private IP2 server to develop it. Is that correct?
I think that's correct. Yes.
Okay. Yeah, that's we were considering making a web interface for ProTurn. So, but I think probably the best way to reach the widest audience would probably be use IP2 because it's already a popular tool, and then it'll be most convenient for our own researchers because they use IP2 for as a search engine.
Yeah, I think that makes sense. I think there's a few. There's actually 2 people in our lab that already developed their own plugin. I don't think they wanted to make it public because in any cases there were pretty specific analyses that they were trying to do that there was not any real interest outside of their outside of the scope of their own experiments. But you can definitely reach a much wider audience if you integrate it with a tool with IP2 potentially. Yeah, that seems like a great idea. But again, Robin is definitely the person that can provide you more details about this. I unfortunately did not develop any plugins for IP2 yet, so the details, the greedy details of the process are a little unknown to me.
Okay. All right. Thanks. Yeah, we'll contact him, get into more detail on it.
Okay.
Thanks. Okay. So then another possibility is Pride and ProteomeXchange. I just got complications. So right now, Pride and ProteomeXchange, they accept data that's linked to publications, and the type of analysis that you would do with ProTurn is generally what you would do before a publication. Currently, there's already a planned collaboration as part of the upcoming reverse expression interface, which is a planned interface to link several different analyses tools together. That'll likely in the future link the repositories Pride and ProteomeXchange with IP2 and COPaKB and other analysis tools. And then other collaborations. So last week when we brought this up, we talked about BioGPS as a potential collaboration. It'd be helpful for people to be able to see different analyses side by side. If we have a web version of ProTurn, it should be easy to integrate that with BioGPS. Does anybody else have anything to add for possible ProTurn collaborations? Oh, okay. Yaset, are you online?
Yes.
Oh,
Can you hear me?
For the Pride collaboration with Pride and ProteomeXchange, is there anything? Is there anything you'd like to add? Any way you see connecting ProTurn with Pride and ProteomeXchange?
Well, we are just starting the API and the expression interface. I think the best way is that when we release this, the first prototype of the expression interface, all of potential tools inside the BD2K can be able to download data very quickly, reanalyze the data, and then perhaps to send again the data back to ProteomeXchange and Pride, especially Pride. But we are just, that we are just starting the first prototype of the expression interface.
Yeah.
In the context of the BD2K project. I don't know how easy or useful it could be for for those tools. We already have our API available, where you can basically see all the experiments and the data, the raw data MCM, the PICLIX files and the results. And we already have some tools like Prime Inspector that enable to visualize this data. But I would prefer to do that in the context of let's say the the expression interface.
Yeah, definitely. Yeah, I know. We know that's still a ways away right now. All right. Thanks. Does anybody else have anything to add? All right. Thanks. I'll give it back to Vincent.
your speakers. Thank you, Brian. So today, Howard, Troy, Tevik, Dinser, and I will propose a few collaborations for COPaKB. And I'll start off with a brief overview of COPaKB so that we can quickly get into the collaborations. COPaKB is a tool that allows you to analyze proteomic datasets that are of or relating to the cardiovascular system. It provides a large-scale spectral analysis as well as high-quality cardiac proteomic data and also relevant cardiovascular phenotypes. So I'm going to briefly talk about our current collaborations. I talked about the GeneWiki collaboration 3 weeks ago. Dr. Ping's lab is working closely with Dr. Su and his team to populate wiki pages of proteins or genes related to the mitochondrial proteome. And in addition, we'll be embedding these GeneWiki articles into our website that will provide users with more information about a protein or gene. And the COPaKB tech team has also been in contact with Cassette, who's leading the proxy or the proteomic expression interface project in developing a simple way of exchanging protein expression data using distributed repositories. So the main goal behind proxy is to support researchers by making it possible to for a distributed search of protein expression data through resources like Pride, Massive, and COPaKB. And the technical teams have had several meetings to start defining the schema, and hopefully we'll give an update of the proxy project in this coming round. Today we'll be proposing a potential collaboration, hopefully with PSEA-Quant and Reactome. And basically we're proposing a disease enrichment analysis tool and known as PPICardio. So I briefly mentioned this tool a couple weeks ago, but last weekend we decided to create a prototype at the first BTK hackathon that our center sponsored. I'd like to thank Dr. Su's lab for organizing this great event. I had a great experience and thought it was very well planned, and a lot of us who attended from Dr. Ping's lab had a great time. So now Howard will go into detail about PPICardio.
Yes, so the idea of PPICardio is to integrate resources from 2 different communities to expand the scope of a protein and cardiovascular disease association. The resources such as ID2, MINT, or INTEC in the International Molecular Exchange Consortium provide information of a physical interaction between proteins, which is called a protein-protein interaction. On the other hand, the resources such as online Mendelian inheritance in man, or OrphaNet, or PharmGKB deliver gene-protein association to disease. So databases in both communities put a lot of effort to update, maintain, and distribute their resources. But unfortunately, each community appears to remain isolated from the other community. So in PPICardio, we try to integrate resources from 2 different communities through APIs to help the cardiovascular research studies. So a user story behind our development is that as a cardiovascular researcher, you ran a mass spec for the samples from heart failure patient and controls, and you identified LRSM1 from the differential expression analysis. And you try to annotate the LRSM1 and no known association to the cardiovascular disease found, but you found that the LRSM1 has a physical interaction with MYL2, which is known to be strongly associated with cardiomyopathy. So we believe this scenario would help to facilitate understanding of novel biological insights from proteomic data set.
Hi everyone, this is Tevfik Dinsir. I'm going to talk a little bit about the technical aspects and how this tool is used. So we put this tool together last weekend during the first BDTK third Network of Biothings hackathon organized by Dr. Su's lab, which was a great event in all ways. So let me just get down to it. So the user inputs a list of proteins in the as uniprots, and those are sent to the server, and the server connects to three different APIs: the uniprot mapping service, the intact interaction database through the Psychic API, and the online Mendelian inheritance in man API. And we get a table of results, and the table also contains information about which proteins and genes are actually related to cardiovascular disease. This is the main screen. This is what you start with. You enter your proteins here, and you get your results. So the ones that are annotated with a heart next to them are cardiovascular diseases, and the ones that are not are non-cardiovascular diseases in this case. There's also the option to display phenotypic series only. In this case, you're only going to get cardiovascular diseases, and you're going to get a higher level of disease families in there, so that you can adjust your granularity when you're making your analysis. When you click on the disease to protein table button here, it's going to take you to this screen, which is going to give you a disease, a protein, and a p-value. And this p-value represents the question, does the list represent the proteins with a specific cardiovascular disease more than would be expected if this were random? Of course, we have to work on this analysis even more. This was a prototype version, but we believe this tool can become useful for researchers in the long run.
So I believe for the cardiovascular disease enrichment analysis, which is to calculate the p-value, I think definitely we can incorporate PSEA-Quant. So in current method for this enrichment analysis, we utilize a hypergeometric test, and it's similar as a binomial test, but difference is when you sample, it samples without a replacement. So it's basically identify which subpopulation are overrepresented in a sample. So for example, to calculate the statistical significance to the cardiomyopathy, it takes 4 parameters. Number one, the number of total genes or proteins in a pool. Second, the number of genes or proteins known to be associated to the cardiomyopathy. In this case, 76 genes were known to be associated. And 3, the number of input gene proteins to the PPICardio. So basically, the genes, proteins in a sample, and number of genes, proteins in the input that are directly associated with cardiomyopathy. So if you don't consider the protein-protein interaction, we found 2 proteins are associated with the cardiomyopathy. If we consider the protein-protein interaction, we found 6 proteins are associated with the cardiomyopathy. So when you calculate the p-value with the only direct association, you get 0.4097. And if you consider protein-protein interaction too, then you will get a 0.027. As Matthew, who developed the PSEA-Quant, as he stated, the limitation of this approach is that it assumes gene-protein expression are independent, and it does not consider gene-protein expression or abundance. So we truly believe we can incorporate the PSEA-Quant to have a higher, more accurate enrichment p-value.
May I just ask you a few questions on your previous slide?
Can you? Hello?
Yes.
Yeah, can you hear me?
Yes.
Yeah.
Okay, so just a few things regarding the details of your calculation. The number means I would be careful because that actually depends on the type of analysis that you're doing. So if, for instance, we're talking about a proteomics experiment, we are not capable up to now to detect all 19,000 proteins that we believe are expressed in human cells.
Yeah.
So the background that you're using can be usually depending on the type of experiment, can be basically the number of number of proteins that you have identified total, and that will take care of having a better modeling of your background.
Yeah, I totally agree. And then, like, the input would be only the differentially expressed protein.
Right. That would be, so that's one of the type of experiments. Exactly. So you have your background, which is all the proteins that you've quantified, and then the test that the set that you're testing is the one that are differentially expressed. Right. Okay. Perfect. I just want to make sure that was something you've considered.
Yeah. I totally agree. Thank you.
Oh, Robert, excuse me.
Oh, yes.
The total number of genes that you are taking into account is 19,000?
Yeah, for this example, yeah.
But is that the number of genes, proteins that you have annotated or considered in your database, or is that, I mean, do you have all of them in your database when you found this 76? Is this 76 out of this 19,000? Is what you have, or is what you assume?
So 76 is actually from OMIM, and I put the total number of approximated number of genes.
No, but I think what you have. I think that what you have to do is to change this number of total genes that you assume that is what exists in humans by the number that you database, the ones that you have, the total number of annotated genes and proteins in your database. Otherwise, the view is the ratio that you are calculating, this 0.004, This is the ratio, and this is wrong because you are not comparing against everything. You are only comparing against what you have.
Yeah.
So you should be calculating the ratio related to what you have, not what you assume there is.
Yeah.
Only to the known world.
Howard, this is Ding. I have a quick question regarding this 76 genes. Can you hear me?
Yes.
Hi, Howard. Where this 76 gene came from?
So it's from various resources. Main resource would be OMIM.
I see. I'm just curious because, for example, if there is a gene within the mitochondria and it will cause mitochondrial dysfunction, in the long term, it may cause some cardiomyopathy, which lead to the disease relevance. So since we know that the mitochondrial genome contains so far, I will say 2,300 proteins. I mean, are those genes could be potentially considered as cardiovascular relevant?
So, Ding, this is Pepe.
Yes.
The 2,300 mitochondrial proteins are not entirely coded by mitochondrial genome. Mitochondrial genome only contributes 13 genes for the mitochondrial proteome plus 4 tRNA splicing components. The rest of the mitochondrial proteome are encoded by the nuclear genome. But I do agree with you, even though there are only 17 important molecular identities in the mitochondrial genome, those should be included. The concept Ding has is absolutely correct, because all 17 of them are crucial.
I think that's, my question sort of,
Thank you, Ding.
Thank you. Howard.
Edward also had a question. Read it out.
Okay, so Edward also has a question. Could you clarify in the null hypothesis, are we consider the possibility that a random set of genes is linked to neighbors that are associated with cardiomyopathy? So basically, the null hypothesis in here is just whether this number could possibly generated by the random or not.
If I may add on this aspect, I think that by using the proteins that are interacting, I was trying to wrap my head around this. I think that you are violating one of the basic assumptions of the hypergeometric test, which is that the associations would be independent from protein to protein, and because they're associated, they are more likely to be annotated in, let's say, the OMIM database. They are more likely to be involved in the same disease, I believe, just because we had some prior knowledge, and so they're more likely to be studied, for instance, regarding a certain disease. I know it's probably not the case for all of them, but it definitely is for some. And so I think that by doing this, you may be artificially under evaluating your p-values, and think and maybe showed some things that are significant that really shouldn't be. I think it's a little tricky in that part as the value of the p-values, if it's really accurate in this context. That's just a point I wanted to make.
Yeah. Thank you. So we would like to incorporate the PSEA-Quant. So it definitely consider the weighted sampling, and I think it's totally, and it also consider the expression level of the abundance of the protein too. So I think it would give accurate p-value. For the collaboration, we'd like to have a discussion for the integration of COPaKB and PSEA-Quant. And we think we would have two options. First option would be to incorporate the PSEA-Quant through the API. And as far as we know, the PSEA-Quant doesn't provide the API. So if we would like to go this direction, maybe we could help to build the API for PSEA-Quant. Or the second option would be we can directly implement the PSEA-Quant to the COPaKB. The second point we would like to discuss would be the velocity of the analysis. So PSEA-Quant uses a Monte Carlo sampling, and this procedure takes time. That definitely depends on the number of sampling. But so, is there any chance to increase the speed of this enrichment analysis? That would be our second point of the discussion.
In terms of increasing it so that it runs and that you click and you get a result in a few seconds, is that your question?
So sometimes it takes more than a second, right?
And it does, definitely, and it can take hours.
Yeah. So my question is, if you increase your power of computing, then do you think we could have a better speed for the analysis?
It would require significant improvement in terms of computing power. My question is more, what would be your expectations? So right now the system is made so that the user submits his query, and then it sends him to a page, and you'll get your results later. At the email you submitted, you'll get the link to the results of your file, blah blah. You can move on and do some other stuff while this is computed. Were your hope to just get that answer a little quicker in terms of a few minutes, or in terms of having the web server, you click and then you get the result almost instantaneously?
So we expect the instant result.
I think this is impossible.
Yeah.
It would require several-fold of speed increase in order to get a reasonable p-value in an almost instantaneous time. So I think that for that part, I don't think it's possible. For now. It might be possible in the future, but not for now.
So I have a naive question. It's Pepe here. So all the computing process, is that done on your server? I'm trying to understand the process. So we submit a query, and it goes to your server for the computational process?
Yes, you're right.
So the server is
You're right, exactly.
hosted at EBI right now, right?
No, the journals.
I'm sorry, at TSRI.
Exactly, yes.
Okay, so this is where the other collaboration we were hoping to explore. I'm not sure where it's possible at all. You know, one of the UCLA investigators on the BD2K team is Professor Jason Kopp. He's the director of NSF Computational Center called Center of Domain-Specific Computing. So part of his expertise, as what he offered for several times, is to improve a specific either algorithm or cluster-based calculation process and to enhance the speed. And he's done it for some of our colleagues here at UCLA genomic institutions, where he was able to shorten some of the analysis process from 24 hours to an hour and a half, is what I was told. And so since he is part of BD2K and he'd like to explore possible collaborations if you guys are interested.
Definitely. So I think you're bringing a very good point. The analysis of PSEA-Quant right now is somewhat optimized for speed, and it's not using the best machines in the world either. So it could benefit from having better.
Well, yeah.
Right,
so,
exactly. So my question originally, when I asked Howard what was his expectation in terms of speed, I think that what you're suggesting, to have an analysis that goes from 24 hours to, let's say, an hour and a half, is very possible with PSEA-Quant. We could bring, so PSEA-Quant analysis takes on average, if I look at the data, it takes on average about 3 to 4 hours. We could definitely bring it in the order of let's say 20 minutes or 30 minutes with an increase in terms of computational power, although it would still be an amount of time that would be long enough that we cannot expect a user, in my opinion at least, we cannot expect a user to just click and have the window to wait until the process is over, until it gets the result. So that's why I was trying to see where Howard was what was his expectation in terms of integration for speed, and when you reach a level that the user has to wait a few minutes, and I don't think it will be possible to bring it down, even though we have access to these. I'm not saying I don't want to do the collaboration with the group at UCLA. I think that's a great idea that we should explore, and I thank you, Pepe, for that. I just don't think that we're going to be able to speed it up to the point where somebody would go in COPaKB, click some button to launch PSEA-Quant, and get the result right after. That to me, with the Monte Carlo sampling approach, seems impossible.
So, but you've very smart pointed out that it could go down from 3 hours to like 20 or 30 minutes, right?
Exactly, which would be very beneficial.
Yeah, that's also that would have been amazing elevation from the current computational time required. Yeah.
Definitely.
So should we get a discussion going with? Is it okay that in the next cycle we invite Jason to give a talk about the specific thing he has done for the genomic world, and see if any approach we could borrow from his group to collaborate on some of these analyses.
I think that'd be super useful. I'd be very much open to that.
Awesome. My understanding is he has a combined approach of both software and hardware. I'm not knowledgeable in the particular area, but my understanding is he could also add a small box. That box is in the size of maybe 5 inch by 5 inch by 1 inch. That plug into that box onto the hardware system also substantially speed up the computational process.
Okay. No, I think it'd be very interesting to hear about what he has to say and what he can bring to the table.
Sorry, can I make a question here? Yes, it. Here. You hear me?
Hello?
Matthew, can I make a quick question?
For sure.
Okay. I'm taking a look to the PSA system, and you basically have an input that is basically one protein, and then the spectral count 1, spectral count 2, spectral count 3, ratio 1, ratio 2, ratio 3. How I mean, because for integration, I always have the feeling that we need to, have something that basically can be, can be integrated between different systems. How much depend of this software, of the input of this software of your data? I mean, what do I need to do before going to your system? It's just a quick question.
I'm not sure I understand your question. The only thing that PSEA-Quant, so basically, I think the COPaKB, well, the system that was just presented, where you submit a list of protein identifications, for instance, would be basically the same for PSEA-Quant. It's just that in addition, you provided abundance value, whether it's in the form of abundance ratios. So if you're comparing, no matter what method you're using, you're comparing two different conditions and you want to make a ratio of the abundances, or simply a more, if I can put it between quotes, an absolute quantification such as spectral count. So you simply, for instance, if you did an IP and you have a list of interactors for a given protein and you have 3 replicates of it, you would give the spec counts of the proteins for these 3 replicates and see if there's an enrichment for a certain Go annotation, or in this context for a certain cardiovascular disease that is among the proteins that are the most abundant in your IP. Am I answering your question properly?
Yeah, sure. Thank you.
Okay. The interesting aspect. So I guess one of the points that Howard was bringing in his previous slides, thanks for navigating as I'm saying this, it was so either to incorporate the PSEA-Quant API. So indeed, there's unfortunately no API for it because of the lack of time. But the option of so I think that there's the option of directly implementing in PSEA-Quant in COPaKB should actually be straightforward if we basically link the 2 web services together. So somebody could submit his data to the COPaKB server, and therefore the COPaKB server would call PSEA-Quant, and that with the submitted data, and that would actually be fairly easy to link the 2 services. And I think we could do the same thing on our side in terms of integrating the COPaKB analysis inside PSEA-Quant. And I think this would help maximizing the impact of both of these projects. If we have access to both of these, we could access to potentially different communities that we would not have access to otherwise. And the part that interested me the most is that my understanding is that you've merged annotation databases of that include cardiovascular diseases, which I would imagine requires quite a bit of time because of all the issues. I guess some of the diseases are not called the exact same way and everything. And I think that's an important resource that can be very beneficial. And I would very much be interested to include the analysis in PSEA-Quant at that level. So I think we can benefit from integrating both analysis and both tools, basically, in terms of reaching a broader audience.
Yeah, I totally agree. Okay, to finish my slides, so the expansion of the scope will be we would like to incorporate the community who studies post-translational modification, also the pathway, and we also would like to engineer the more sophisticated disease image analysis tool, which is we would like to employ the PSEA-Quant. Yes, thank you.
Thank you, Howard. Do we have any questions for Howard or anything about the collaborations that he proposed?
So I guess I'll ask another question to Howard. So when you're thinking of integrating, does the idea of basically having one web service being the bridge to the other one something that you had in mind, or you'd rather really integrate the entire analysis so that it runs on your servers? I don't have any issues with one or the other. I think the fact that if we just link the two query service together and that the execution remains for now on each server can be done very quickly and and be available in the next following weeks, maybe even. So that might be an easier thing to do first, or if you were thinking to fully integrate it on your own servers.
I don't see. So I don't think our goal is to house everything at UCLA. And it's not, number one, it's not possible. Number two, I don't think it's necessary. So your proposal of linking the two resources, having that as a first step going, and then let's see what would be the sort of user requirements after we implement that. That's something I would propose.
Great, I agree.
Are there any other questions? Okay, if you have any other questions, feel free to email me or Howard, and we can continue our discussions for our open discussions either after this meeting or for our next conference. But that about concludes our discussions today. I'd like to remind everyone to sign up for a discussion slot for this second round. If you have any questions about the sign-up form or anything related to the conference, please feel free to email me or Brian. And also, as I noted at the beginning of the conference, we will be taking a 2-week break to allow everyone to sign up for a slot for this coming round and have a set schedule. Thank you all for attending this conference.
Thank you, everyone.
Thank you.